Okay, so let's start a little bit about me. So I grew up in the southern archipelago here in Gothenburg on the island Styrsö. If you have not been there, wait until summer and go there. It's very beautiful. For my studies, I moved into town and did my bachelor's in chemical engineering and physics. Then I proceeded to do my master's in complex adaptive systems, and then my PhD in the biotechnology department, now life sciences. And now I'm currently in a postdoc, so I didn't move very far in my studies, perhaps after this postdoc. Okay, so in my PhD, I have dedicated the whole work to this biomolecule, the proteins. And before I started, I thought that the proteins are mainly in meat and in protein powders that you buy in the store. But of course, proteins are these amazing molecules, and they are built up by just 20 different amino acids. And they are stringed together in these long polymers chains. And these polymer chains then fold into these very beautiful structures and amazing structures. And it's really this structure that gives the proteins all of their different functionalities, all from enzymes catalyzing different reactions, all the way to huge protein structures that give structural features like your hairs and your different tissues in your bodies. And so proteins really make or facilitate life as a whole, and all life depends on these molecules. And recently there has been a lot of useful things in industry using enzymes and proteins, all from in the food industry to pharmaceuticals, and one often neglected part is detergents. So, and as I said, it's really this structure of proteins that give the function. And this structure is highly dependent on the environment that the proteins are in. And here is one of the examples for temperatures. So as temperature increase, these structures tend to be unstable and fall apart, and thus the enzymes or proteins lose their functionality. So, and this will become more important later. Oh. Okay, that's... So it's just these ones? Okay, that's a shame. It worked fine on my computer before starting. Okay, so let me try to explain what it's supposed to be here. So, as I said, recently a lot of enzymes have seen uses in industry. And of course, since these are biomolecules that are developed by life and evolution has shaped them, they are not always applicable directly in an industrial settings. So usually we do have to do some engineering to make them work. And how this has been done previously is in the lab, where we start out by a natural protein as a starting template. And then we introduce a lot of mutations into these sequences, and we get a huge variant library. And from this variant library, we test it again in the lab and see whether or not we gain the functionality that we want. And then we select that, the best sequence, or the sequence with the highest fitness, and we do the same procedure again. We introduce mutations and we test a new variant library. And as you can see, that's a very time-consuming process. So what we wanted to test. Did we? So, okay, now, okay. Okay, so what we tried first then, can we split this up into something that we can instead do in the computers? So first we tackle this part where we introduce mutations. So it's called the diversification step. And we wanted to use generative AI to do this step. And just a quick reminder for everyone, nowadays everyone is aware what generative AI is since of the recent hype. But just as a reminder, so discriminative models are these where we are trying to construct a decision boundary between classes in a feature space so that we can classify between different distributions in our data. Meanwhile, generative models try to learn the distribution of the data itself, and so that we can sample from these distributions of data. And nowadays we have really good models that can generate these beautiful images just by inputting a prompt. We can even guide them towards what we want in the end. And okay, so and what we wanted to do then is to learn this distribution of natural sequences, and so we employed these strategies, or similar strategy here to learn. Instead of images, we learn sequence distributions. And one of the
I think technicians, I think we will have to unplug it and do just this presentation just for the people in the audience, so that we can see the image. It works on the Mac, but it doesn't. Don't want to miss out on these beautiful illustrations. There we are. Let me also, now we don't have this, but could you?
Yeah, we will do it the old-fashioned way. Okay, so this is the protein engineering cycle, where we start with a parent sequence, we do some diversification, introducing mutations, creating these variant libraries. Then from the variant libraries, we do some experiments to evaluate the fitness of the sequences. We select the variant which has the best properties that we want, and then we most often need to repeat this cycle many times. And so it's very labor intensive. And here, and yes, so instead we tried to learn this diversification step through learning the distribution of natural sequences using a generative machine learning method. And of course, the value in this is that we can now, instead of experimentally introducing mutations, we can, and also in a unstructured way, we are introducing these mutations in the experiments. Here we can use a generator then that is trained to sample from a natural sequence space instead. And we can sample as many variants as we want. And actually in our lab we were one of the first to experimentally validate that we can de novo generate these sequences in the machine. So that takes part, or that takes care of the diversification step. Now then, the really labor-intensive task is actually the selection part, where we're doing the assays in the lab. So what about this? Can we do something for this? And remember I said one of the properties of this, the activity of the proteins are really sensitive to the environmental factors. So, for example, temperature. But we also know that life exists in very harsh conditions, not only in room temperature and in the lab conditions, but all the way from frozen Arctic to hot springs, where temperatures can reach above 100 degrees. So we thought that maybe we can use this data and to train on. And actually, what we can see is that experimentally this is very difficult and labor intensive. So there is not a lot of data that pairs sequence data with this temperature measurements. So instead we, and of course this is not new for or specific for biotechnology and proteins, but a very common occurrence in real life when we're doing machine learning. So a common technique is that we are using transfer learning, and where we learn or we teach the model on a data set where we have or an adjacent task where we have more data. And here we can actually, we actually do have this kind of data set where we, instead of taking the experimentally validated proteins and how temperature affects them, instead we can use what's called optimal growth temperature data, where we measure and label the data with where we take the organisms that contain thousands of sequences instead and label them with where the organisms grow. And doing this, we instead get millions of sequences instead of just only tens of thousands. Yeah, and even though this is a sort of more coarse-grained label, we are able to our models are able to capture these variants in this data. And of course, the real task then is where the optimum temperature for these enzymes, and we are also able to increase the performance of these models. using this approach of transfer learning. Yeah, so then we can select for this property where, or thermal stability of the proteins. But we also really want the proteins to be active and functional for the task that we want it to do. And this turns out to be a very difficult task as well, because just a single mutation in a sequence can actually destroy the protein and make it non-functional. And here again, I show you the image that we generated before. And of course, it guides it towards the prompt that we want. This is indeed a dog standing on the moon wearing a Christmas hat. But we are also seeing there is something wrong with this image. And there is, in fact, a moon in the background. So which moon is it, really? So this is this kind of small artifact in the generation that I'm talking about. So indeed, the properties are there, but it's still wrong. So, and at this point, there was coming out these foundational models in protein engineering. So think of it as the ChatGPTs, but for proteins. And we thought that maybe, since it has seen such a large proportion of natural sequences, maybe we can use these models to sort of recognize whether or not the sequences are real, or if they are indeed containing some artifacts. So, and what we did is we took two of these foundational models and we used them to score these sequences. And what we could see is that we could increase by 50% of generated sequences to also have activity in the lab, which doesn't sound that much, perhaps, but it's actually a game changer since it's so labor intensive. Okay, so now we have diversification with the generative models. We have the selection parts, both with our trained regression models for the property and the scoring methods for activity. Can we now then put all of these together into one system so that we are able to engineer a protein in the computer instead of going around the loop in the lab? And this is what we tried to do next. Again, we tried to make a generator to generate sequences or sample sequences from a distribution or a slightly narrower distribution where we are also generating toward a specific property. And usually when we have a task like this, it's really nice if we have a lot of paired data where we have both one that is less stable and then one that is a pair that is more stable. For proteins, this is not usually the case. Instead, we have a bunch of unstable proteins or less thermally adapted, and then we have a few that is thermally adapted proteins or thermophilic. Again, in computer vision, we already have solutions for dealing with this style transfer or property transfer. So can we do the same for proteins? And indeed, we tried similarly as before to learn the distributions of thermally stable proteins coupled with these foundational models to also sample more directly from the functional parts of the protein space. And indeed, and then coupled with the regression model to increase selection even further, we were able to generate sequences that had as much as almost 40 degrees higher melting temperature, or temperature where the structure breaks down. And of course, it's also nice to not only do an experiment once, but we actually showed that for a majority of sequences generated with our approach, we were able to increase the melting temperature. And yeah, lastly, I would just say that yeah, science is not done alone, and I'm very grateful for the lab that I've been working in, and also for the whole division that we are a part of, systems biology, and of course Chalmers and everyone here. So, thank you.